Structure of blood vessels. Composed of 3 layers, tunics. Tunica intima, composed of simple squamous epithelium. Tunica media, sheets of smooth muscle. Contraction, vasoconstriction. Relaxation, vasodilation. Tunica externa, composed of connective tissue. Lumen. Central blood-filled space of a vessel. Structure of arteries, veins, and capillaries. Structure of arteries, veins, and capillaries. Figure 19.1a. Arteries carry blood away from the heart. Types of blood vessels. Arteries carry blood away from the heart. Capillaries, smallest blood vessels. The site of exchange of molecules between blood and tissue fluid. Veins carry blood toward the heart. Elastic arteries, the largest arteries. Types of arteries. Elastic arteries, the largest arteries. Diameters range from 2.5 cm to 1 cm. Includes the aorta and its major branches. Sometimes called conducting arteries. High elastin content dampens surge of blood pressure. Figure 19.2a. Muscular, distributing, arteries. Types of arteries. Muscular, distributing, arteries. Lie distal to elastic arteries. Diameters range from 1 cm to 0.3 mm. Includes most named arteries. Tunica media is thick. Unique features. Internal and external elastic laminae. Figure 19.2b. Types of arteries. Arterioles, smallest arteries. Diameters range from 0.3 mm to 10 micrometers. Larger arterioles possess all 3 tunics. Diameter of arterioles controlled by local factors in the tissues. Sympathetic nervous system. Figure 19.2c. Smallest blood vessels. Capillaries. Smallest blood vessels. Diameter from 8 to 10 micrometers. Red blood cells pass through single file. Site-specific functions of capillaries. Lungs, oxygen enters blood, carbon dioxide leaves. Small intestines, receive digested nutrients. Endocrine glands, pick up hormones. Kidneys, removal of nitrogenous wastes. RBCs in a capillary. Figure 19.3. Network of capillaries running through tissues. Precapillary sphincters. Capillary beds. Network of capillaries running through tissues. Precapillary sphincters. Regulate the flow of blood to tissues. Tendons and ligaments, poorly vascularized. Epithelia and cartilage, avascular. Receive nutrients from nearby CT. Capillary beds. Figure 19.4a. Capillary beds. Figure 19.4b. Capillary permeability. Endothelial cells, held together by tight junctions and desmosomes. Intercellular clefts, gaps of unjoined membrane. Small molecules can enter and exit. 2 types of capillary. Continuous, most common. Fenestrated, have pores. Structure of capillaries, cross-section. Figure 19.5a. Structure of capillaries, cross-section. Figure 19.5b. Routes of capillary permeability. 4 routes into and out of capillaries. Direct diffusion. Through intercellular clefts. Through cytoplasmic vesicles. Through fenestrations. Low permeability capillaries. Blood-brain barrier. Capillaries have complete tight junctions. No intercellular clefts are present. Vital molecules pass through. Highly selective transport mechanisms. Not a barrier against oxygen, carbon dioxide, and some anesthetics. Wide, leaky capillaries found in some organs. Sinusoids. Wide, leaky capillaries found in some organs. Usually fenestrated. Intercellular clefts are wide open. Occur in bone marrow and spleen. Sinusoids have a large diameter and twisted course. Sinusoids. Figure 19.5c. Conduct blood from capillaries toward the heart. Veins. Conduct blood from capillaries toward the heart. Blood pressure is much lower than in arteries. Smallest veins, called venules. Diameters from 8 to 100 micrometers. Smallest venules, called postcapillary venules. Venules join to form veins. Tunica externa is the thickest tunic in veins. Mechanisms to counteract low venous pressure. Valves in some veins. Particularly in limbs. Skeletal muscle pump. Muscles press against thin-walled veins. Figure 19.6. Vessels interconnect to form vascular anastomoses. Organs receive blood from more than one arterial source. Neighboring arteries form arterial anastomoses. Provide collateral channels. Veins anastomose more frequently than arteries. Tunica externa of large vessels have Vasa Vasorum vessels of vessels. Tiny arteries, capillaries, and veins. Vasa Vasorum vessels of vessels. Nourish outer region of large vessels. Inner half of large vessels receive nutrients from luminal blood.